As a reminder for folks, please use the chat feature if you have any questions or comments during the presentation, and I will read off the questions and comments to Wade at the end, time permitting. So Dr. Wade Wilson is the Assistant Director for the Southwestern Native Aquatic Resources and Recovery Center, and he'll be presenting today a virtual tour of the center and its capabilities and facilities. And Wade, whenever you're ready.
Going to be giving a kind of a background information on Southwestern Native Aquatic Resources and Recovery Center. We're located in southeastern New Mexico and Dexter, New Mexico. And then I put together a virtual tour, kind of a 45-minute video that has some text, and I'll be talking as we go through it as well. So I'll be going back and forth between some slides and the video. So just give you an idea of where we are. You should be able to see a map right now. We're about 23 miles southeast of Roswell, New Mexico, and like I said, southeastern New Mexico. And we're just east of Dexter, New Mexico, and Lake Van, if you know where Lake Van is. So let me start the PowerPoint, or stop the PowerPoint and start the video. And can you see the video now?
Yes.
Okay, so let's start this. So welcome, thank you all for coming down. And maybe if you wanted to, rather than sitting all day, stand up and walk around with me during this video in the virtual tour. So in the background there, you can see some fish culture and some storage buildings. This is the main admin building that has both research and the admin offices, which includes the director, a center director, which is Manuel Ulibarri. I'm the deputy center director. Trista Becker is the fish health lead, and then we have some admin people. So we have three main programs. Let me just show you our general outline of how we're set up. We are basically have fish culture, research, fish health, a maintenance program, and an administration program. And I'll go into each of those as we go through. And fish health is also called aquatic animal health. So we were established in '31 as a fish hatchery, basically warm water species for sport, such as catfish, bass, and we've gone over several name changes, but with the passage of ESA, we basically all deal with everything that's threatened and endangered now. This is the research program that was started in 2001, and at that time, genetics was the main focus. So let me share what the research program is. So basically we have genetics, pathobiology, physiology, and within genetics, we do conservation genetics, invasive species, early detection, eDNA. Within the pathobiology, I'll show you the lab. We have molecular diagnostics of basically fish health cases, and from time to time, emerging pathogen issues using genetics. And within physiology, we have reproductive ecology, histology, and in the past, we've looked at stress responses. So these are the, you should see the video, is that correct?
Yes.
Yes.
Okay. Sorry, I keep asking. So this is the freezer room. We have tissue samples of individuals that were collected back in the 80s. So we have both extracted DNA and tissues that have been archived from species all over the nation that we work with. And we archive them here. And then we also work with University of New Mexico Museum of Southwestern Biology and we provide tissues to them, and then we ask for tissues from them as well. So it's a good collaborative program there with them. So this is the physiology and pathobiology lab. So on one side to your left is the physiology, and on the other side pretty much is the pathobiology. So we have a couple of reproductive ecology projects we're working on using Colorado pike minnow on Rio Grande silvery minnow. And looking at, with Rio Grande silvery minnow, the reproductive ecology to look at temporal egg development in the species using histology. And let me just show you some slides. So basically you should be seeing histology techniques, correct, a slide. So basically most histology is used paraffin wax, but it wasn't hard enough to hold eggs, and so we could slice through eggs. So we developed this resin technique. It's a harder material that can be used for bones, and that's basically what it was developed for. We tried it on eggs, and we can get these good crisp sections. So we use this microtome. We embed the tissue, in this case ovaries, in the resin, and then take slices. We stain them and mount them to slides, and then taking samples from various time points, we can look at the eggs and egg development and see, try to get an idea of when they're gearing up for reproduction. So back to the virtual tour. So this is the microtome that I just showed you, where we slice everything. And then pathobiology is basically the aquatic animal health program using genetics to look at various species and pathogens. Within research, this is one of the pieces of equipment that we can use to select different DNA fragments. And this is usually used for next generation sequencing because we only want fragments of a certain size. We don't want everything in the genome. And then this is the area where we do extractions of DNA and setting up of polymerase chain reactions. So each person, staff member, is assigned a hood. This prevents contamination. They have their own pipetters. Each hood has UV lights to decontaminate the area after each workflow activity. So this is setting up a PCR. And Melody right now is putting in a master mix. So this is basically all the reagents for the chemical reaction. It has all the primers, all the enzymes, everything we need. And then you just add a fixed amount to each well. It's a 96 well plate that she's working with. And then she's basically trying to figure out concentration of primers and all the different reagents to have the reaction go well and come off, providing clean data. She ran it before and they really didn't work as well as she wanted to. So she's going back and trying to do different concentrations. So each of those tubes she's pulling has a different primer set in it, essentially. So has everything else in those tubes is the same except for the primers. And pretty much everything we do in genetics is pretty much this. You mix a bunch of chemicals in tubes and then you pipette them to plates and then you add the DNA and you put them on different machines. So now that all the reagents are added, then the next step is to add the DNA. And this is the DNA working plate. So we extract the DNA and then we take a portion of that and put it on a plate. And we know which individuals are in each of the wells. She's only going to be using 8 individuals to do this test. So she'll use the same 8 individuals in each of the different reactions, each of the primer reactions. And this project that she's working on is basically to look at the genetic diversity and parentage of wild Colorado pike minnow in the San Juan River. It's something we've been working on for a number of years with the San Juan program and ES. We have some good markers where we can really start looking at parentage and how many individuals are reproducing in the river and that sort of thing. Other projects that we're working on in the lab are broodstock management, Gila trout, patchy trout. We provide them. And then we're looking at freshwater population structure in Texas, freshwater mussels. This is the main molecular ecology lab. So this is where a lot of the equipment is that once we set up the PCRs, we put them on thermocyclers to copy the DNA, and then we put them on machines like this automated DNA sequencer. And this allows us to analyze different fragment sizes, such as microsatellites, and then sequence specific regions of DNA, such as mitochondrial genes. And this is used fluorescent technology. So each of those primers that I talked about are labeled with different colors. So when they pass, go through this machine, they pass in front of a laser and light emits in certain wavelengths, and we can tell what it is. This is older technology, so you can only sequence one gene region at a time. We're transitioning to next generation sequencing. This is an Illumina MySeq next generation sequencer. So you can sequence hundreds to thousands of genomic regions of a sample at a time. So it's much quicker, and you can get a much bigger chunk of the genome to look at. And one of the applications we've used that for recently is to basically do the sequence the whole genome of the mitochondria region for various species in the Southwest. And I said we can contrast genomes of different species or individuals across different areas. And then we can also look at selection of particular genes across environmental conditions and look at local adaptation. So these are thermocycler machines, thermocyclers. So that well that you saw, or that plate that you saw being worked on, you put it in here, it heats up, it cools down, and allows the DNA to be copied. You start with 2 copies, one from your mother, one from your father, and you end up with millions of copies at the end within several hours, going through 20 to 30 cycles of the PCR reaction. So behind you, this is a agarose gel station used for various things. Basically, you're looking for something to occur or not occur. Basically, you do a PCR, and then you pour a gel. So it's being done here. And then this is a fish health case. Basically, what they're looking for is a particular pathogen. They have species or strain-specific primers that they used in their PCR. Now he's pouring a gel, and it's in its liquid form. And this has dye in it that comes with the gel mix. So basically binds to the DNA and allows us to visualize it. This is him placing a comb. So when the gel solidifies, there'll be these little indentations or wells that can be used to load the samples, and then electric current will be run through it. And then the DNA will migrate through the matrix. Again, once the fragments have been separated on a gel, we can put it in this box, close it, and visualize it, and then take images with a camera and then view it on the computer. This is a quantitative real-time PCR machine, so this can be used for eDNA, which I'll talk about in a little bit, and then basically you can look at how many copies of a gene you have or fragment size at each stage of the PCR cycle. And this is a bioanalyzer. With next generation sequencing, it's really, really important to have really high quality DNA. So this allows us to quantify how good the DNA is before we can do any downstream applications. So that's research in the main building, the administration building. So we're going to go into the maintenance here in a little bit. So that's the maintenance building, and to our left is the fish culture building that we'll go into a little bit later. So we'll walk over in here and see the other piece of research equipment. A few years ago, we installed eDNA clean room. You can see some of the ponds in the background. We'll get to those as well. So we as I said, installed a eDNA clean room. Because of space requirements, we had to put it in maintenance. But this is, as we'll see in a little bit, 3-room clean room. And this is the rest of maintenance that we'll see here as well as we walk through the building. So right now, what we just passed, we'll have a wall, and all the researchers will come through that door. They'll pass through the samples in this box, so there's not any contamination of when they're gowning up. Here in a little bit, we'll go through the entry room of the clean room. And these have HEPA filters, and so it basically keeps all the rooms separate and all the minimizes any contamination with projects. So they'll gown up here with lab coats, put on lab coats, and then there are foot baths that these will be filled to protect against contamination and biosecurity, so you don't bring other pathogens or DNA or anything with you. And there's the sample box, so you can get your samples and bring them into this room to prepare the samples for the next room. We're still trying to get situated. We thought originally that this room would be where we filter the samples before doing DNA extractions, but we're getting everything on filters as it is, so that may be an extraction room, and this is where the PCRs are set up just like the other room in the other building. So they have dedicated pipettes, UV lights, again, minimizing contamination between projects if there are several researchers that are working on it. And this is a one-way trip through the clean room. And then once you're finished with the project, you can't go into any other area. You have to get rid of your lab coat and leave the clean room before working with anything else. This is the rest of maintenance building. So we have a wood shop, lots of stuff going on in fish culture and maintenance. This one thing that's done in the woodworking shop, as you'll see, is we use a lot of boards to control water flow in the ponds. So we make those boards, we plane them because we want a tight fit to really prevent water from going through the boards and keep it in the pond as much as possible. And there's always lots of plumbing to do, so we have a nice organized plumbing room that we can find parts whenever there's a leak or something that needs to be repaired or replaced. And we have a couple of offices. Typically, maintenance staff, we've had 2 to 3 people on staff as far as maintenance goes. This is where we can do welding and repair vehicles or trailers. That's a transport trailer, small transport trailer there. And then a vehicle lift if we need to raise something up. That's a vehicle storage building in front of us. And then we'll pan to the right here in a second and see some ponds. Those are 10th acre ponds mostly that were put in for experimental purposes, but with the bringing the silvery minnow on board and a few other species, they've been converted to grow up ponds for production. Here are some boards and screens that were in ponds when we drain them. So we bring them over here and pressure wash them and clean them up before they're moved to the next pond and used. So we'll go into the vehicle storage. And then some Kubotas that we use to transport feed and some other things from pond to pond, rather than having people walk everywhere. It's fairly large area to get around. So as far as fish culture, we have multiple hauling tank options. We have 1, 2, and 3 compartment trailers that can go either on the bed of a truck or on a trailer. This happens to be a 2 tank or 2 compartment hauling tank. And this allows us to separate fish based on locations where they're going, if they're going in different locations or if they're different sizes or different densities. This is a 1 compartment tank. And then we also have, I believe, a 2 compartment tank that goes in the back of this truck as well. And then oxygen bottles while we transport can be tied to the back of either the trailer or the back of the truck to keep oxygen flowing as we transport. And then various water tanks, forklifts, mowers, so general storage in the dump trunk. There's always roads to repair and add gravel and move things around. So there to your left is the maintenance building. Fish culture is right in front of us. This is just another example of hauling tank. This is a 3 compartment hauling tank. So there's 1 here on the side and then 2 in the rear, like some of the other tanks that we have. And then we can attach hoses to those and then let the fish out as needed. And the recipient. So this is a heavy equipment storage building. And you'll see some ponds back there, concrete raceways in front of us. We'll get to those in a minute. And this is, again, the fish culture building. This is a water line to fill hauling tanks, either while we're on station going between ponds or if we're going off station to transport fish and stock them. So we have 4 concrete raceways for fish holding, sorting, tagging. So we'll harvest them, bring them into these raceways and do what we need to do. From time to time, we also can do those activities indoors that we'll see here in a little bit. We do have a refuge population of humpback chub that we have kept in these so we can control temperature and not have them reproduce. So the grooves in the concrete walls allows for replacement of like what we see in the boards and the screens. This allows us to subdivide the raceways as we needed. This is a similar situation to what we have in some of the ponds where we can add boards and screens to control water flow. And these are the larger ponds that we have. We have a total of 77 lined and unlined ponds that range from 10th acre to 1 acre. So these are bonytail. They're in the lower Colorado. And then 1981, we collected the last 11 individuals. They spawned 5 males and 5 females. And we have the only broodstock with a backup broodstock in Mora, New Mexico. And they basically have to go out above 305 millimeters. So we have to measure them before we put them out. They're all pit tagged before they go out. So we scan them and then document both their measurement, how large they are, and their pit tag number. So those 2 are associated. If they're not, as it said earlier, large enough, then they're pulled aside and put out in the ponds for further grow out. And then this database is given to the partners so they know exactly what pit tags went out, what individuals went out, what their pit tags and numbers were, and what their size was. So now that's the vehicle storage. Now we're going into fish culture. This is the fish culture building. And it's, as we'll see, it has 3 different bays and an office area. This is the office, 2 of the 3 office areas. We have another little office area off one of the bays. And we have 77 outdoor lined and unlined ponds, and this is where fish culture keeps track of what is in each of the ponds. They have codes for each species, what year class they are, if they're broodstock, wild caught, refuge, or production. A lot of those when I filmed this were empty. So there are times when they're all full, and there are other times when some of them are empty right before we stock everything out. Again, foot baths for biosecurity. 3 bays. This is the East Bay. We have a Middle Bay and a West Bay. And various size tanks. So in the winter, we bring in a subset of some of the smaller fish species to just protect against winter conditions. As they brought in, they have a card on the side of each tank about, what species they are, when they were brought in, what pond they were brought in from. This is Guzman, beautiful shiner in New Mexico. It's extirpated from the US. It's found in the Guzman Basin of Mexico, where it still exists, but we have the only refuge population. It's being considered for reintroduction down in the Membres area in southern New Mexico. Big Bend Gambusia from Texas. And then some of the other ponds back there are holding, I'll talk a little bit later, some Colorado pike minnow we brought in from the wild. And those tanks also can be used for, if they're empty, and available for research purposes where you can do replicate experiments. And this is desert pupfish, one of our other smaller species. Right now, I believe on station we have 14 different species that we manage and maintain. And some of these, like the desert pupfish, are refuge populations where we really don't do production or stocking. Others, like the bony tail, are really large production commitments where we produce every year and stock. So now we'll go over to the Middle Bay. So again, fish culture building. And we have these long raceways where we can bring in individuals for a variety of activities, whether it be spawning, sorting. These are the silvery minnow. Each of these 10 tanks that you see there are filled with silvery minnow that have been tagged. So this is the 2020 year class. So we found that to minimize stress after each handling event, we salt the water and let them rest for about a week before the next handling event. So if they're going to go out and be stocked, we bring them in and let them rest for a week before we handle them again, whether that's tagging or putting into a tank for stocking, like I said. These are razorback sucker, young of the year. You can see in there those various sizes. So this is the time of year where we're stocking a lot. So a lot of the tanks are filled and things are being prepared to go out. To your right, there's a tank there where after equipment is used in a tank, it's decontaminated before it touches another tank or goes to anywhere else. Again, these are all 2020, 10 tanks of 2020 year class Rio Grande silvery minnow. This is a room, tuna room. We have egg trays in here for hatching. And each species is a little bit different. Some are kept in raceways or aquaria, and some are used in the egg trays. Again, another foot bath before you go into another room. This is the West Bay. Some waders to be used when people go out into ponds or raceways and they need to get in. Some aquaria, some different tanks. As we go down, these round tanks in the end are where Rio Grande silvery minnow are spawned. And once they spawn, the eggs are collected and put into the aquaria on the right. And once they hatch, eventually the larvae are transferred out to the lined 10th acre ponds for grow out. Just some bigger raceways for larger species or larger ponds. Again, letting them rest before handling activities, or you can sort based on size, tag, prepare for spawning, that sort of thing. Razorback sucker. Which occurs in the Colorado River and the San Juan River. So it's one of the Colorado River kind of long-lived species, which includes humpback, razorback sucker, bony tail, and Colorado pike minnow. So just a basket you can put in one of the raceways, and if you're sorting, you can put fish in there or put smaller amounts of fish in there to deal with and handle rather than a huge raceway. These are some gravel baskets. So some species require gravel or crevices to spawn. So we can provide these in smaller, this is experimental species that we're working with, trying to figure out how to get it to spawn in captivity, but we can also provide those in ponds to have them spawn out in the ponds as well. And this is another area where we can attach spawning jars or aquaria to handle eggs or fish. So we've seen this, that's the inside of fish culture. So now let's go out to a 10th acre pond and see how it's harvested. So these are the 10th acre ponds, as I said, that we put in. So this is the lined pond. It's being drained so we can harvest the fish. What you see in front of you is the kettle. There's lots of algae in there. So basically the pond is sloped, so the water flows from the other end towards the kettle. As we remove the boards, we can control how fast the pond is drained. And then as it's draining, we clean out the kettle and we get out all the debris and algae. So when the fish eventually get into the kettle, they are clean and they can be harvested without getting all the algae, and they don't suffocate. So they're cleaning the screen and pulling out all the algae. As last amounts of water are going down into the kettle, some of the fish they don't want to really go into the kettle, so they have to be encouraged to go in, and this minimizes loss from fish getting stranded that like to hang out. So now that all the fish are in the kettle, it's left for a while with water flowing to temper the water. So it matches what we have in the trailer and inside. So now the fish culture is going to seine. the kettle. And again, these are Rio Grande silvery minnow, the ones that we saw in the building before. And there's a little concrete barrier around the kettle, and this is sloped as well, so all the fish go in through that front little entrance. And the water, they're not stranded on the sides. And you can see how dirty, how much algae and debris there is in the pond. The pond will be dried, and then before it's used again, it'll be swept and cleaned and prepared for the next batch of fish that come into it. And as you can see, there's minimal algae and debris because it was cleaned before. So these will be carried up to a transport trailer. Once all the fish are in the trailer, it'll be driven to the fish culture building, and then the fish will be transferred indoors or to one of the concrete raceways, whatever species is being worked on and what raceways and tanks are available. Again, this, you can see there's a net inside the trailer. It allows us to easily get the fish out of the trailer into nets and then transport to tanks. So that's, they've now driven to a fish culture building. You can see there's an oxygen tank on there that allows oxygen to be provided to the fish while they're being hauled in the tank. And just to our right is all the concrete raceways that we saw before. So we're at the back of the fish culture building. And these will be moved to the middle bay where we were earlier. So yeah, these are the tanks that we saw earlier. Now they'll be placed in those, and then you can see bags of salt that will out here in a little bit. Again, reducing stress after a handling event. And harvesting is a very stressful situation. So there's the salt being handling or added, again, to mitigate stress. So now let's move to a different building. This is the quarantine building, or a quarantine room in a building that also has some fish health labs. So anytime we bring something on station, it goes into quarantine isolation for about 30 days while they're treated for internal and external parasites. And then we also mitigate stress. So it's a lengthy process. We want to deal with stress and some of the external parasites first. And then once they're doing well, then we can start treating for internal parasites. These tanks were just cleaned, so the water levels are a little bit lower. But these are Colorado pike minnow caught in Utah. We're trying to build a new breed stock. It's a long-lived species, and then they reproduce. They're sexually mature from about 6 to 10 years old, so we have to collect them years in advance before we actually need them. This is a multi-year, multi-agency effort. We have one more year really to get our numbers. So we're looking to collect about 1,000 individuals from the Green River and 1,000 individuals from the Colorado River in Utah for our purposes and for stocking into the San Juan River. And then because they will eat each other, we'd have to do separate based on size. So this species takes up a lot of space on the station. And really it takes years before you can really combine them and they won't eat each other. These are Z habitat units we can use these for experiments where we need replicates, either different temperatures or chemicals. You can change out the size of the tanks. They have a heater, they have chillers. These are really nice units for research. And again, we've used these with the San Juan program, looking at different temperatures. So now we're going to look at fish health. This is right next to the quarantine. Fish health is in two different buildings. This is the necropsy room and part of the room where they look at parasites under microscopes. So fish are either shipped to us, or they go out to a site and collect tissues and then bring them back. But if fish are shipped to us, which has happened a lot with COVID-19, they are dissected here and all the tissues are collected. And we'll see that here in a little bit. And then anything macro parasites that they find can be examined under the scopes for identification. Now we're going to go into the virology room. And then basically samples prepared and placed on various cell lines, and then they're incubated at different temperatures and examined for lysis. So basically there are cells, and if there's viral activity, you end up with a clear spot on the plate. So you know there's some sort of viral activity going on. Otherwise, you see the lawn of cells is uninterrupted. And they use different, the cell lines are developed from different fish species. So there's another area in the virology where the samples are prepped. So this is basically where samples, once the tissue is ground up, they pull the supernatant off that has the viral particles in it and leave behind the tissue. We'll see here in a little bit. And then put on the, this is also where they're put onto the different cell lines. So this is an inspection, various species. So staff are collecting tissues as far as part of an annual inspection. So the federal facilities have to be inspected at least once a year before anything goes out, and then they're certified that they're clean, free of pathogens, and fish can go out. And you can see that they're in groups of 5. This allows pulling of different tissues for testing. So you can actually pull some of the tissues and just test whether or not the group of fish was positive or negative. You don't have to do each individual. Some tests they do on individuals, like bacteriology. Others, like virology, they can pull a group of individuals. So it's just collecting tissues. I believe these are going to be pulled for virology testing. So here we are in the lab. So as I said, they're put into a solution, the tissue is put into a solution and ground up. And this is where they try to clean up the samples. So they just have a nice supernatant that has the viral particles and then all the other tissue that might degrade the cell lines that they're placed on. The leftover tissue doesn't degrade those cell lines. So their supernatant is pulled off, they're spun in centrifuge, and then the supernatant is pulled off again. And then once the samples are cleaned up, they're placed on the cell lines, which are the plates there on the left. And I believe each one of those has a different cell line. So each of these vials that he's working with has 5 individuals in it. So in total, there are 60 individuals that are being worked with, which is the typical lot for aquatic animal health, fish health, annual health inspections. They looked at 60 individuals per lot. And this pipette allows him to tell the pipette how much to release at the push of a button, so he can just, the right amount will be put onto the plate at each pressing of a button. And once those plates are filled with the different individuals, then they'll put in the incubators, and then each cell line has a different, I believe, a particular temperature. So this is the second building of fish health, aquatic animal health. This one has staff offices, parasitology area, and a bacteriology area. And this is a dark room where they can use scopes and use it on particular tests where they need to look at, I believe, fluorescence in a dark room. So with salmonids, they look at whirling disease or look for presence of whirling disease. The area we're coming up on our left here, that's where they digest heads. So it's in the cartilage of the head of salmonids. So they have to digest those and then look for the actual pathogen. And the rest of this is mostly bacteriology. And we'll see some people working here in a moment, but this is the general layout where you have some hoods where you can work with samples and clean them out so you don't, again, get contamination between different sets of samples. So the way you identify bacteria is you run through a variety of different tests, and then you see if they're positive or negative. And this, what he's doing right here is putting some bacteria on slides to look for motility, and he'll put them under a scope. And this is a test that has, or strip, contains 20 tests. In each of those little wells, you put the bacteria, the sample in there, and there'll be a chemical reaction. And you can determine using a key whether they're positive or negative. And then you add up all the positives. And then going through a key, it'll determine what kind of bacteria it is and whether that's a reportable pathogen. And again, if there's any question about what it is, they can always grow it out and take it over to the genetics research area and sequence it, or use primer-specific probes, species-specific primers, and run it out on a gel to see if it's positive or negative for that pathogen of interest. So that's it. Thank you for watching and paying attention. So there's the virology. Straight in front of us past that storage unit is the virology area, the quarantine. At the very end is actually a feed room that we didn't look at. And this building on your left is the rest of the bacteriology offices and parasitology. And that's the entrance road coming into our, from our right. You can see some of the ponds, kind of where we were when we looked at harvesting and the 10th acre ponds. And we're back at the entrance of the building. And that's it. That's the virtual tour. I hope you've enjoyed it.
Okay, thank you so much. So I see we have one question that popped up from Catherine Murphy. She commends your camera skills, Wade. She says it's a fascinating tour of a very impressive facility. Looks like you do all of your own or most of your own maintenance. How self-sufficient is the facility?
It's fairly self-sufficient, but we do a lot of contracting as far as well maintenance. When we put in new kettles and liners, we also contract that work out. So we maintain pretty much everything, but a lot of the big construction projects and drilling of wells, chemically treating wells, we contract all that work out.
Fantastic.
Do you want to add anything? I believe William and
Manuel are on, and I've made them both co-hosts so they can
Okay.
unmute themselves.
And then Trista Baker as well. She's the
Okay.
fish health lead.
I will let her unmute.
And I don't know if Steve must have been called in.
Yes, I do see him.
Okay. Yeah, he's the research unit lead, so that we can field all the questions.
All right. Are there any other questions for Wade? I'm going to let folks type in their questions. We've learned yesterday that folks type slower than they can speak. So what is I do actually have a question for you all. If you had a wish list for additional capacity for your facility, what would it be?
I think a lot of the requests we're getting are from, they're coming out of the San Juan program, is to, rather than creating more fish to stock out, we're considering how to provide better fish. Mm-hmm. So we're doing, exploring ways to flow train. So we've taken some razorback sucker that have gone into the San Juan River. We flow trained them in one of those concrete wasteways, I think for 3 to 4 months. And we gradually increased the flow and then put substrate in there to break up the flow. So they had some quiet areas, and then they had some areas to swim around. and play in the flow. And they actually did kind of quickly went back and forth, and they played in the flow, and then they would rest behind some cinder blocks. So stuff like that requires, if we're doing a lot of it, some of the numbers that we put out in the San Juan are, you know, tens of thousands, if not hundreds of thousands of fish. We need a lot more pond or raceway kind of facilities that need to go in, and then wells will have to be drilled to provide water.
Mm-hmm.
So that's one way we're looking at increasing infrastructure. Do you have anything to add, William or Manuel?
Yeah, this is Manuel. Thank you for having us on, and good job, Wade. I agree with you on the capacity when we're talking about potentially trying to increase the number of fish that are going out, or doing a hatchery enrichment on those that are going out to help improve their survival. I think the major need that we have, and we have been discussing this with our fisheries division, is dedicated space that's specific to the research activities that we do. And what I'm talking about is we're constantly having to adjust our program to be able to conduct some of the research that we do, just because there's not enough facility to have a dedicated space solely for the research. So it's in a bit of a conflict every now and then when we're trying to do production and we're trying to do research. So we've submitted, actually Wade has submitted a request to our engineering division, this was a while back, to try and get a dedicated building to conduct all the research activities that we'd like to do. I think that's the biggest, would probably be one of the bigger needs on the tech center side. And of course, on the hatchery side, we're maxed out in terms of the physical plant. We would have to build new infrastructure, as Wade indicated.
Okay. We had two more questions come in from Casey Ish from MRGCD. He's asking, why is this facility in Dexter, New Mexico? Is there spring water there that is unique to that area?
So yeah, we have wells, deep wells that we pull water on, and then we also use city water that's chlorinated. I don't know about the chemical makeup of our well water versus, it's really hard. So it's probably quite a bit different chemically than some of the other areas. William or Manuel, if you want to, you know more about the chemical makeup of the water and how it compares to some of the other rivers and areas we stock.
Yeah, I think I can field some of this. So we end up using about 1,600 acre feet a year, which is about half of our current water right. We're pumping off of a shallow water aquifer in the basin here. We're not essentially tapped into any artesian wells or anything like that. The water is extremely hard. Parts per thousand in terms of total hardness is going to be anywhere from 2,500 to 3,500. A little bit high in salinity, in chlorides, which doesn't necessarily hurt us in terms of fish culture. And the hardness has not historically caused any health issues, nephrocalcinosis, anything like that, based on the HAI or health index that we do on the fish, as well as fish health inspections. So yes, it is quite a bit different how we try and work with that when we're stocking all these other locations throughout the Southwest, is to do a slow temper, not only to equalize water temperatures, but to also try and equalize, or at least add some of the river water, wherever they're going, the receiving water. We actually temper for water chemistry as well. So we're not immediately shocking the fish going from one water chemistry to another. Tempering time, depending on water temperature itself, as well as location and time of hauling, could last from a couple of hours to 6 to 7 hours, just to try and equalize the fish's osmotic balance as they're being moved from our water to the receiving water.
Okay.
And then to answer the question about eDNA, I think Grace had that, or someone had that. So far, we've had 2 eDNA projects. One that we conducted before we had the clean room, where we worked with Bitter Lake National Wildlife Refuge, looking at spring snails and their different water areas. And then one that we've picked up since we put in the clean room, which was only a couple of years ago, working with this, some facilities in Arizona, looking for presence or absence of New Zealand mud snails outside of one of our hatcheries out there, federal hatchery that raises various salmonids. They found it in the river, and they wanted to know if it was on the station and how far up and down the river the spring snail